Thank you, Mindy, for inviting me to this conference. And the title that I was given, and maybe we could turn the lights down a little bit, thanks, is Computer Robotics Interface, the Caltech Experience. And so just to start off, for me, the experience has been a great one. It's there are very few other fields like it where it requires such an interdisciplinary approach and work across many institutions. And in particular, our work here will be the advancer doesn't seem to be working. Okay, there it goes. Maybe my battery's low. It's really amazing to work with such a large group and requiring people from a variety of different disciplines. So this includes people with medical backgrounds, scientific backgrounds, engineers, just a whole variety of expertise. And yeah, I think this is why I couldn't change it, as my battery's running out of the clicker. Maybe, Ted, you'll be my automated clicker. From our lab, oh no, please go back one. Thank you. From our lab, we have, or maybe if I get closer, if that'll work. From our lab, we have several people that are here today. Christian Klaus, Kelsey Pasha, Spencer Kalis, Ying Shi, also Marcus Hauschild, and Yuri Ninja. And so probably you might have already been talking with them during the breaks. Also, this project, since it's quite an extensive one, in large part is funded by DARPA. And one of the DARPA representatives is Kent Pankratz, who's right over there. And yeah, actually members of the lab, could you raise your hand too? Great, okay, so hopefully in the break you'll be able to talk to them and get more detail about the project. And the DARPA project is referred to as Revolutionizing Prosthetics. And it has a number of different universities and groups collaborating. We have from Applied Physics Laboratory, a group led by Mike McLaughlin, and they've designed, to my knowledge, the most advanced prosthetic limb that is currently available. At USC, we're working with Charles Liu, who you saw talk last night, and who will also chair our panel. He's a neurosurgeon. And actually, we got started with USC through Brian Lee, who is a fourth-year neurosurgical resident, and was a PhD student with me when he did his MD PhD degree at USC. And then also Christy Heck, who's also here. She's the neurologist that's collaborating with us. At Rancho Los Amigos, of course, Mindy, who organized this conference, also Kathleen Shanfield, who's been helping us in recruiting patients, and then a large group of therapists and other staff here at Rancho. And then also included in this project and things I'll be telling you about is UCLA. You heard Bita mentioned BlackRock. Actually, it had been the company originally started at Brown, now in Salt Lake City. We're also collaborating with members at the University of Chicago and also at the University of Pittsburgh. Again, Vito mentioned the clinical study that recently was performed there that is part of this revolutionizing prosthetic project. Now, it works. I'm close enough. Maybe I'll try it from here again. So basically, the idea is if you have lesions of the nervous system, like a spinal cord lesion, there are parts of the nervous system that are still active. And so if you place an implant in the brain, then you can extract those signals and use them to drive an external device that's used for assisting the patient. And no, it's okay, I'll just get closer. So we record neural signals from the brain, and it's an array of electrodes, as Vito was mentioning, about 100 electrodes per chip. And then in our case, we decode these signals and use them either to operate a robotic limb or a computer tablet. Now, these, most of the studies we do first or so far have done in monkeys. These are two of the monkeys that participated in this research. After they usually work in the lab about 5-6 years, kind of like graduate students. Then they get, after that we take the gear off and we retire them to animal sanctuaries. The graduate students aren't so lucky. They have to go out and find jobs. Now, I mentioned this limb. They refer to it as the modular prosthetic limb, and that's because and it's very nice, actually. So it's modules, and so you can have, for instance, just the hand if you have an amputation of just the hand. From our point of view, it's advantageous because when the monkey uses it, and even humans, sometimes you break off a finger or something like that, APL will then send you a new finger rather than having to replace the whole hand. This limb has 17 degrees of freedom, so it's very advanced. In this particular case, the Matt here is wearing a data glove, and he's here you can see some of the degrees of freedom. The hand is very dexterous and can handle soft objects, and even, like in this case, not crushing the water bottle. Can use tools, and here's an example of it manipulating a clothespin. So now this is an example of a monkey operating the MPL. In this case, again, with the data glove on his hand. So he's reaching out. You can see there's the monkey here with his hand controlling and his job is to reach out and squeeze the ball. And if he squeezes the ball, then he gets a drop of juice reward. So here you can see he's squeezing it, and then he stops and drinks his juice reward, waits, and then up comes the ball again, and he proceeds. Now, you can see, comparing him to the human, he's a little more impatient. But Yuri has been training him, and now he's much more well-behaved and will go from ball to ball in a less impetuous way. Now, part of this project, as I mentioned, is also colleagues at the University of Pittsburgh, and they implanted a patient, Jan Schuerman, who had cerebellar spinal degeneration and was is paralyzed. And this study was done with Andy Schwartz and Mike Boninger and Jen Collinger, who you'll see talking here. So she'll first introduce the patient, and then you'll see the patient, Jen, operating the same MPL limb.
From the beginning, we've been really focused on, how much control can you get of this arm, moving the hand and wrist and everything, but, Jan's goal has really been to feed herself a piece of chocolate.
In this case, there were 2 implants in the motor cortex, so they were a pretty large number of units, and so they could get a lot of degrees of freedom in the movement of the limb. Now, these experiments, and also the ones that Vita was talking about out of Brown, have been recording from the motor cortex, and this is the part of the brain that sends out the final execution signals to make a movement. In our clinical study, we'll be targeting a different part of the brain. This is the posterior parietal cortex. And this is an area that forms the initial plan or intent to make a movement. And it then sends that intention to the motor cortex and premotor areas where it's then elaborated into the execution signal. Now we would like to take advantage of this more cognitive signal, so we refer to this as a cognitive prosthetic, because we think in some ways it will be more intuitive for the subject and also in some ways more efficient. This one nice aspect of the coding of signals in the posterior parietal cortex is there's a very strong representation of the goal. So usually in the motor cortex, you get a representation of the trajectory, so it's giving you information about moving the limb along a particular line of movement. In the parietal cortex, you get a very strong representation of what you want to do. It sort of says, I want to reach out and touch this particular object. So this is an example of the intent signal in the parietal cortex, and see if you can tell which way this little popping of the cell is intending a movement. So it's like a plan that's being put in place. So how many people think the cell was coding for the left? Good. Yes, that's right. And so we record from a large number of these cells, and in a test we have the monkey think about moving to a location, but not actually moving, and then we give him a cue about where we think he's thinking about moving. So, oops. So the triangle will come up and tell him where to think about moving, and then it'll come back again, indicating where we decoded where he was planning to make a movement. And the delay here is about a second. We record activity for a second, but within the first 100 milliseconds, we can already decode the goal of the movement. So it can be extremely fast. It can be, actually, we can move these cursors faster than a person could, because otherwise it has to go through all the other neural structures and eventually up to the spinal cord and muscles. Also, most of the research in this area requires actually making a movement to a target that's flashed on the screen. And of course, a lot of our movements are not made that way. We don't have things always flashing up and indicating that's where we should reach. Usually we're reaching in a rather stable environment. So here's an example where the arrow tells the monkey where to position the cursor. And so, and that little click indicates each time he does a successful trial. So so we refer to that as a symbolic coding. So that was great. We have a representation of the goal of the movement. But recently we found that the parietal cortex also, and to the same efficiency as motor cortex, can also code the trajectory. And we think the reason for this is not that it's generating actually a raw copy of the motor command, like it would have in motor cortex, but rather it's what's referred to as a forward model. So that's a feedback of what the motor cortex might want to do, which then informs the parietal cortex and provides information about the state of the limb in real time. And it turns out you can also generate these forward models just by imagination. And there's numerous lesion and other studies that suggest that this forward model exists in the parietal lobe. So we think we're actually reading out the trajectory signal that's used for a different reason, but is very similar to what you find in motor cortex. And so this is an example now of a monkey in virtual reality, and he's reaching to locations in three-dimensional space, and we then use this information to decode the trajectory. And this shows what he's actually seeing. And this is actually in 3 dimensions, so you can kind of you lose the 3rd dimension. And in the corner is the, you can see the monkey reaching out there in space. And here's an example then when we switch to brain control, decoding the trajectory. And he's still moving his limb, you can see, even though so he thinks he's moving the ball with his limb, but we're really actually using his neural signals. And in this case, then, we trained him to generate the trajectory signal without moving his limb. So you can see that he can just think about making a movement and generate this forward model for the movement and generate the trajectory without actually making the movement. So when we, in these studies, you can see that the feedback is a visual signal. So the monkey knows how he's doing based on what he sees. But patients with, say, that are deafferent and only have vision, so when they go to manipulate an object, they do very poorly. In fact, they seem almost like they're paralyzed because they don't get the feedback from the sense of touch and proprioception. And so, and just to give you kind of an example of this, when you go to the dentist and you get an injection of Novocaine, you feel like you can't talk because you're not getting this feedback anymore about the movement of your mouth. And this is really extreme if your entire limb is deafferented, as it would be with a paralyzed patient. So one additional and important source of feedback would be somatosensation. And both our group and the Pittsburgh group are working through the Revolutionizing Prosthetics program to provide this feedback signal. So normally we'll decode the neural signals to move the limb, but then the hand, this APL is doing the sensorization of the hand. So they have a large number of sensors on the hand, and then that information is fed back to another array of 100 electrodes that's implanted in the hand representation in area 1 of the somatosensory cortex. And so the stimulation then will provide, sort of close the loop to provide somatosensory feedback to the subject so they can improve their dexterous movement. And so this shows an example. We've done this, made these implants in monkey, and here's a monkey in reach control that's now controlling a virtual limb to these locations. And here's an example where we're controlling the limb in what's called a handbag task. So the animal can't actually see the target that he plans to reach to, but he gets stimulation when he's like reaching in a handbag to find an object, and he gets stimulation in this hand region, and then he has to hold his hand over that location. So he doesn't see these little arrows here. Those are for yours, just for illustration, so you can see where he holds the hand. And he's also doing this in 3D, so you can see that the surface doesn't always exactly line up with the actual 3D location of the limb. And we've extended this to a case where the monkey feels an object, he sees an object and feels the texture based on the frequency of stimulation. Then he reaches into the handbag and gropes around, and he has two objects in there which have different frequencies of stimulation. So it's like feeling the roughness or the texture of the objects. And then he can identify those particular objects. There's also an area in the posterior parietal cortex, AIP, which codes the grasp of the hand. And in this case, you can see that this particular neuron favors a particular type of grasp. So this is very interesting because you can, with a single neuron, get the entire grasp configuration rather than having to rely on a number of neurons coding the particular locations of or desired locations of each finger. And another application that we're using is to control a computer tablet, and this is using both goal and trajectory signals. So again, the idea here is that you have so many applications out there in, say, on iPads and Androids, that a paralyzed person could really do an awful lot just by using all these applications. And so this shows an example here. Usually in these tasks, you've noticed that a light is flashed and the monkey moves to a location. But if you're operating on a screen with a lot of different applications, you have to sort of match your internal representation of the particular icon to the ones you see on the screen. And so here the monkey, this is under brain control, gets a sample, and then he has to match it to the different search and find the correct icon to hit. And you can see that he can do this really fast. It's nice because it's operating with a bunch of distractors, so it's not like just flashing a target and then suddenly going there, which is a very simple task. Here, you actually have to search around to find, so he has to identify the cue, move the cursor with his brain to the cue, then go out and find the appropriate match. And you can see that he does this awfully, very well. I don't know if I can do this that fast on my iPhone. So we're now beginning human trials. So we've obtained the regulatory requirements for that. We've begun through Rancho to recruit patients. We've also, and this is working with the USC Medical School and Rancho Los Amigos, for the robotic control with the stimulation feedback. We're to the point where we're about to submit an IDE to the FDA for this, and hopefully that will go through, and we'll be able to, in the near future, also do this kind of implant in human trials. So, thank you.